Good morning, everyone, and welcome to today's class. I want to welcome all of you, and I want to believe that you have no problem in the audio. Kindly confirm before we start the class at 7. Kindly confirm that the audio is clear and that you can hear the lecturer. You can please type on the public chat on YouTube and confirm. Simply type confirm audio clear. I'm requesting that you confirm audio clear in the YouTube link in the YouTube channel. Good morning and welcome. As we wait for our time and as we wait for others to join us. So kindly let me know from the YouTube channel that you can hear me, that you can that you are receiving the audio clear and loud. Thank you, Shikari Kelvin, Kalimi, Eugene, Faith, Stephen, Justin, Wanza, who else? Douglas, Kabusana, Joy, John, Othiambo. Who else is in the house? Esther Wamboi. Who else do we have? Thank you so much. Confirmed. Motuko, Loiza. We have 21, 526. Gabriel, Dennis, Eric. Thank you, Paul, Matthew, Diana. Who else do we have? Francis, Gideon, Michael. Well, I'm encouraged to see that you're trying to join before time. We start our lecture exactly at 7, and then maybe we are going to have to take around 1.5 hours in the lecture, and then at least we proceed to our respective classes. Thank you very much, Kiprotich, Eugene, Joseph, Achieng, blessings, karibuni sana. Karibuni, and welcome to my YouTube channel where we preach and drink immunology. It's a very interesting subject, and today I want to take you through the immunology of HIV/AIDS. Welcome, everyone to my channel. Well, this is part of the University of Embu, but it's part of my channel, so I want to welcome you again. I'm quite delighted to see that majority of us are trying to log in before time. So karibuni sana, and keep on joining, keep on coming, so that at least and inform your colleagues who are sleeping. Maybe someone is in bed. This is an early morning class, and I know some of you are not very good early risers. So kindly text them, inform them that the class is starting at 7, and we are learning some very interesting stuff about how our immune system works. So inform everyone, the class reps, inform everyone that the lecturer is already here and ready to start at 7. Thank you, Blamwell. Kindly confirm, well, immediately you log in, confirm that you can hear the audio, it's loud and clear, so that the lecturer knows how to proceed. Good. We wait for others and we wait for time. Thank you so much. Just be patient. We start at 7. We start at 7. If you have any concerns, if you have any questions, you can please type in the public chat. If you are close to my office, then you can join me in the office and come and listen directly from my office. My office is next to the clinic, the university clinic. But of course, I cannot hold all of you in my office at this time. Susan, Samuel, Kilunda, please immediately join in. Kindly confirm loud and clear so that the lecturer knows that the audios are working. All right? The audios are working. And again, thank you so much, and karibuni for today's lecture. Evelyn Mwendwa, who else is in the house? Thank you. We are almost starting. I do not know. I can only see that 32 of you are watching, but I do not know how many have already joined. Let me refresh my system. 36 so far. It's 6:59. We should be starting in a minute's time. Inform your colleagues to wake up and join the class. Okay, class reps. Inform your colleagues that we are almost starting the class and everyone should join. Otherwise, they are going to miss very important things. Christopher, welcome. Immediately joining, kindly confirm the audio is loud and clear. 41 of us have already joined. It's 7 a.m. in the morning. And according to the timetable, we should be starting our class right now. And so, ladies and gentlemen, it's my pleasure to welcome you to my class. And in today's class, we want to be learning about the immune system. My name is Julius Dilango, and I am an immunologist, and I have the pleasure of taking you through how the HIV virus interacts with the immune system. I want to believe that all of you will walk with me through the lecture, and we are going to pace ourselves and try to walk slowly so that at least we try to uncover the jargon, the immunological jargon that you may have come across in some of the lectures that you may have had. Karibuni, and I want now to ask that who is willing to pray for us? Or you don't start your meetings with prayers. Well, if John Jogona or Lea Makungu want to pray, they can pray wherever they are, and once they have prayed, they can inform us that they have finished praying. But ladies and gentlemen, today I have the pleasure to introduce to you the immune system as we know it, the immune system. And the lecture objectives in this class, the first thing that I need you to understand is clearly be able to define immunology, what we mean by immunity, be able to name the tissues of the immune system. Number 3, we be able to explain. By the end of the lecture, we should be able to comprehend and explain the cells of the immune system. Again, we should be able to describe the structural components of the HIV virus, because it's the virus that infects our body, and our body have to protect themselves from this virus. And then number 5, we would wish to identify the immune cells that have CD4 receptors, because I want to believe that majority of you have come across this term, CD4. Majority of us have heard either in the news or in the newspapers, we have come across that term, CD4. And so we want to demystify what we mean by CD4. Again, it's important and imperative, number 6, to understand and be able to explain the clinical cause of the HIV disease. How does the disease progression from the point you get infected? All right. By now, I want to believe that we all appreciate that infection from HIV/AIDS come either from sexual transmission, blood contact, and predominantly from sexual transmission and from blood contact. So these are things now that we should hold dear at the back of our mind. All right. Number 7, we should be able to explain how the infection with HIV/AIDS leads to clinical disease. All right. So this is another phenomenon that we want to appreciate, because some people get infected with HIV/AIDS. We have heard stories about people called carriers. All right. People who have the virus, but they never get the disease. Okay. Carriers. And so we want to appreciate how then do the people who get the many diseases that are results from the infection with HIV, how do they get these diseases? Number 8, we would like to appreciate, or rather by the end of the lecture, I would like you to appreciate and examine the events that accompany the symptomatic disease, the symptoms, of the HIV disease. By the time you get AIDS, which are the symptoms that accompany the individual. And then, of course, lastly, we would like to understand the pathogenesis, rather how and why does the HIV virus cause the various symptoms that it causes in the body. So ladies and gentlemen, welcome on board. It's my pleasure to have you. And like I've told you, my name is Muguero Julius Dilango, and I happen to be an immunologist. I teach in the Department of Biological Sciences, and I think I'm interacting with some of you. I'm teaching some of you, especially the guys taking which course? Economic Statistics, Economic Finance, and BSc Finance. I'm teaching them. We usually meet on Mondays in LT01, and you're also free to join my class at Mondays at 3 p.m. Okay, on Mondays at 3 p.m. Remember, at the end of this lecture, again, you are expected to go to your respective venues, and in your respective venues, you are going to meet the lecturers, and the lecturers will have tutorials with you, and of course, explain things that I may have omitted or things that are not clear. But at the same point, I would encourage you, because we are not in any hurry. We are here to learn and interact with one another. I would encourage you that you ask as many questions as possible, especially wherever it's not clear. At the same point, I have indicated some activities. If you go to the online e-learning platform, if you move to the e-learning platform, you're going to find topic. Is it topic 6 or 7? And under my topic, which has my name, you're going to find some e-learning or rather activities that, or some assignments that I've asked you to do. Kindly attempt them. I do not know whether I'm going to be able to collect for all of you, but at least for my class, I want to believe that I will be collecting, and I'll be noting who did the assignment and who never did the assignment. Take the assignments with the seriousness that they deserve. Kindly address them, and make sure that you are at the same, you always do what the lecturer asks you to do. Ladies and gentlemen, to start us with, we would like to appreciate what we mean by immunity, okay? Because this is a common term that we always come across in many of the platforms, or in many things that we hear in our contemporary lives. What do we mean by immunity? And I want to believe that majority of us may have come across that term, immunity, all right, which we simply define as resistance to disease, the ability of your body to defend itself. And so without using so much clinical jargon, I want to tell you that every house that you know or that you build has walls. And the reason why we have walls is so that we can keep away cold, we can keep away hot air, etc., etc. So these walls are supposed to protect us. It's the same case with an individual. It's the same case with a human being. A human being have a skin, they have hairs, they have etc. Even the skin in your body forms part of your defense system. So I want to believe that that's clear, that even the skin that you have in your body forms part of your defense system. It's supposed to protect what is in the inside of you, all right? And so the defense mechanism in humans, or rather in human beings, or in living tissues, or rather in living beings, is comprised of cells. Number one, we have cells, all right? The first component that defends us is cells. The second component, the cells are contained in tissues. So again, we also need to appreciate that these cells are contained. Rather, an accumulation of cells makes tissues. And of course, other than that, above the cells, we have molecules that makes these cells. All right? We have molecules that makes these cells. And so to start us with, majority of us have heard about something. Now that we are living in the pandemic times, we have heard about something called antibodies. All right? Antibodies. These are proteins. These are molecules that defend us against infections. We have heard about cells. We have heard about common cells that are called CD4 cells. These are common things that we hear in the street. CD4 this, CD4 what, that. Or rather what we call white blood cells, all right? White blood cells. All these are part of the defense component. So take note, the molecules that defend us. Examples is an antibody. The cells that are involved in the defense, an example is what we call the CD4 cell, which we are going to define later, and what you may call white blood cells. So take note of that, white blood cells. It's important that we appreciate that. For the tissues, ladies and gentlemen, the tissues are many. You have heard about bone marrow, that thing that you like sucking when you are taking your nyama choma or your chemsha, all right? Even the blood is a tissue, and this one function of the blood is to have a protective role. It is to have a protective role, trying to keep your body away from diseases. And so, ladies and gentlemen, I want you to take note of this, that it is an agglomeration, an aggregation of so many things that make the immune system. And the system, quote unquote, is simply a system, simply a network of the skin, of organs, of cells, of molecules, of barriers, etc., etc. Even the fluid. Some of you ask, why do we have saliva in our mouth? Why do we have mucus in all our openings? If you look at your eyes, we have some fluid in it. If you look at your ears, we have some fluid in it. If you look at the vagina, if you look at the mouth, the nostrils, all these openings have some fluids, and all these fluids also form part of the protective network. So, ladies and gentlemen, by now, I want you to appreciate that the immune system is an aggregation of many, many things that join forces to protect the body against infectious pathogens. All right? So that should be very clear, that it's an aggregation of many things that come together, or rather that work in cahoot to protect the body against infectious pathogen. That should be very clear. We move on to the next slide, where we meet how the cells, the tissues rather, of the immune system are distributed. And as you can see, from a human perspective, geographically, from the head all the way to the legs, these tissues are positioned in such a way that, they are geographically well positioned in all entry points. As you can see, we have something that is supposed to protect you. Okay, starting from our nostrils, our mouth, all right, and even all over the body. You can see from adenoid to the lymph node, to the kidney, to the appendix, to the lymphatics, and all these things are supposed to help us fight infections, even in our digestive system. All right, even in the digestive system, you find that you will find these things that are positioned clearly geographically to help the body fight the infections. Next, ladies and gentlemen, we come across the cells, and rather the tissues, the cells that we find in that help fight infections. So in a nutshell, these are what we call white blood cells. To any person who has not been trained in biology, these are what simply are called white blood cells. Okay, this is using common layman's language, white blood cells. And these cells are rather contained in two tissues, either in blood or either in tissues. That's what I meant. All right. Either you find them in blood or in tissues. What do you find in blood? Simply the aggregation of the white blood cells. And of course, majority of what you find in blood, you still also find it in tissues. We have this cell, the first category here, the neutrophil. The second category, the eosinophil, the basophil, the monocyte, the T cell, the B cell, the natural killer cell, the platelets, and even the red blood cells. All these form part of the protective cells that you find in the blood. If you move to the tissue, again, you still find the same players. Of course, with the addition of a few, like the dendritic cell, and also the macrophage, all right? And also the macrophage, and even the mast cells. So it's important. And again, all these diagrams, we are supposed to draw them. You can retrieve these materials from the resources in the e-learning platform. You are supposed to draw all these diagrams. I believe that I have communicated. Okay, now moving on. So that at least we focus our lecture to simply HIV. We don't just address, we are not simply in a class of immunology. We need now to ask ourselves who is involved or which cells are involved in protection against the virus, the HIV virus. And ladies and gentlemen, it's my belief that majority of us, having grown in the contemporary times that we are living in, and majority of you having been born post 2000, that's what I want to believe, because majority of you are either young or less than 20 years, then you may have come across this term, the CD4. That term, majority of us should have come across it, either in high school or in publications. And I want to emphasize this morning that this term, it's commonly associated with infections with HIV virus. And the term CD4, CD simply means cluster of differentiation. That's what CD, the term the CD means, cluster of differentiation. So this is an immunological jargon that we use to differentiate cells of the immune system, especially the cells that we call lymphocytes, the T lymphocytes. And the reason why we use this cluster of differentiation is because it's very difficult to differentiate between the lymphocytes, the T cells and the B cells. Perhaps it's important you understand this, but that morphologically, if you look at this cell under the microscope, the cell called T cell here and the cell called B cell, it's very difficult to differentiate the two of them. And so using markers, using molecules on the surfaces of these cells called cluster of differentiation, all right, and it has a number 1, number 2, number 3, number 4, very many of them, we are able to identify them using this system of naming called cluster of differentiation. To differentiate means to take an immunological role, to take a unique role, okay, to play a unique role, to differentiate, to become a unique cell that plays some certain role in the defense system. So that's the first thing that I would like you to appreciate, what we mean by the CD4 system. Then the second thing that I would like you to pay attention to this is the CD4 cells. What do we mean by CD4 cells? And what we mean by that is this. This is a group of cells that are called T helper cells. Group of cells that are simply referred to as the T helper cells. So that should be very clear. A group CD4 system simply means a group of cells called the T helper cells. And the job of these helper cells is simply to help, just like the word denotes. Okay, here I've used the word activate, but in actual sense, it's not activate. They help. And how do they help? They help by communicating. They help other cells by communicating. Okay, communication. And ladies and gentlemen, communication in cells or at the cellular level is done by molecules, is done by secretion of molecules, okay? Or rather what we call signals. So kindly pay attention. Anytime you come across the term cluster differentiation cells, it is simply denote T helper cells. Okay, all right. The term is loosely used to mean T helper cells, but in the immune system, T helper cells are not the only cells that have that molecule called cluster differentiation 4. We have other cells that have that, but in matters HIV and AIDS infection, CD4 cells simply means T T helper cells. So I want you to take note of that, because later on we are going to introduce some other cells that also have CD4 cells. So CD4 cells, I want to repeat myself, simply means helper T cells. And helper T cells, their job is to help B cells, to help cells called killer cells, and to help another category of cells called macrophages. And the help comes through communication, and communication in cells is done through signaling, or rather release of molecules. We are not going to go into the immunology of the signaling. Now, having appreciated that, ladies and gentlemen, the next thing that I would like to bring to your attention is the two arms of the immune system. The two arms of the immune system. So generally, when an infant or a neonate is born, they are born with a skin, they are born with a mouth, with eyes, if they are a normal neonate, or if they are a normal infant, and whatever protection that they are born with from their mother forms part of what is called innate immunity. Innate is something intrinsically born with, okay? Something you are born with. It forms part of the innate immunity. This is what we define here in A, all right? Innate immunity, all right? And this provides immediate protection against any microbial invasion. And part of the innate immunity, just to bring you to speed, is anything that you find in the skin, even the sweat that we produce forms part of protection. Okay, proteins, some proteins in the saliva, some proteins in the saliva. Pay attention to that. Some proteins in the eyes, or rather in tears. Let's say in tears. Okay, some cells that we are born with, and what else? You know, a variety of many things, even the mucus that forms many of the opening that we have in our bodies. Mucus, okay? All these form, or rather comprise, what we may call innate immunity. Sorry, what an individual is born with to protect them, all right? Number two, pay attention to the adaptive immunity, which means that as an infant grows, they keep on getting infections. You know, I believe majority of us have interacted with babies, or we have seen babies growing. And if you go to any hospital, the majority of the patients are babies, less than 2 years, less than 5 years. And you are going to find that, number one, they have a lot of mucus on their nose tubes. They cough. They have fevers. They have what else? You know, babies have so many, you know, small illnesses. And these illnesses, they develop these illnesses because they are weak. Their immune system is weak. So immediately they get an infection, their immune system is sensitized, and once the infection is managed, the immune system adapts to that infection. We call that adaptive immunity. We call that adaptive immunity. And the two main players, or rather the main players in this so-called adaptive immunity, are cells that we call lymphocytes. All right? Cells that we call lymphocytes. And lymphocytes, ladies and gentlemen, are simply the cells that we call B cells and T cells. B cells and T cells. Okay? We may take a breather. We go back to the adaptive immune system. I have given the analogy of a young infant, or rather an infant or a neonate. Once they are born with their skin, they are born with their sweat glands, they have eyes, they have mucus, etc. I have simply said that forms part of the innate immunity, what the baby is born with. All right? Then, as the baby starts breastfeeding, blah, 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 and growing, they get infections. and even vaccination, even part of the vaccination to protect them against some diseases. All right. So this baby, as they grow, they become infected with some certain diseases. The diseases are managed. They develop cells. Then these cells, which is still they are still born with, are called lymphocytes. We have seen we have two categories of lymphocytes: the B cells and the T cells. The B cells and the T cells. And these two cells, ladies and gentlemen, are what forms part of adaptive immunity. And adaptive immunity, as we have mentioned, is a slow progression of immune responses, but these immune responses are very specialized. They form a very specialized mechanism of defense against infections. And this adaptive immunity is also what is used in the principle of vaccination. That's why we are given vaccines. What is used in the principle of vaccination, in vaccines. Okay, and I want to believe that we may now be getting vaccinated against the new virus that is circulating around the world, the SARS-CoV-2 virus. But majority of us, the last time that we got a vaccine was when we were infants. We got the BCG vaccine, we got the tetanus vaccine, we got all those vaccines, even the measles vaccine, mumps, rubella, name it. All these vaccines were weakened form of the disease that was put in our body so that the body develops B cells and T cells, and these two cells are supposed to protect you against a future infection. So that forms adaptive immunity, ladies and gentlemen. Is that clear? Now that I can't access YouTube channel, I don't know whether we are okay. I want to believe that we want to take a break, a breather, so that I check what is on the YouTube channel. Okay, and see whether we are doing fine, or rather I'm talking like a preacher. We are not in church. We need to engage and we need to understand what we are doing and why we are learning and what we are addressing. So CCS 103. Okay. So I'm using the same computer. I'm taking a breather now, so that at least I see whether we are on the same page. So from the YouTube channel, I can see that 283 of you are watching. I hope you are not just watching. As you watch, kindly, thank you, Otieno Kennedy, for indicating that I'm crystal clear. Lesson is clear. Mwambia. Victor Kiprangat, ndio kuingia bana. Topic ya leo ni wewe. Victor Kiprangat, you come late and then you start asking what I may call quote unquote. Let me not mention that. But you come to class, you come quietly, you join, and you ask your colleague what we are learning. All right, Rachel, Dixon, Edna, Monica, Faith, Melody, clear, Festus, clear. So as I really do not wish to be so fast, I want you to be making some small notes. If you can, because there are some things that I may mention that are not emphasized properly in the notes. Okay, all right. And yeah, the lesson is good. Teresia, the lecture is quite interesting. Phineas, it's good. Doctor, we are okay. Thank you very much. If you have any question, kindly try and type in, and then I'll try to get back to the public chat. One of my computers is not working, and so I'll be going back briefly. I'll be stopping to take a breather so that at least I check in my laptop. So can we please proceed, ladies and gentlemen? Thank you very much. At least I can see there is participation, but the attendance is not quite good. We expect more than 1,000 students. We are only 274 of you watching. So don't just sit there. Inform your colleague that we have a very interesting lecture and you should subscribe to my channel. And after this, we are going to have an interesting classes. We are going to have tutorials in our respective classes. So inform your colleagues that they should not be late. All right. And of course, immunology is quite interesting. Again, I would wish and pray that you ask the class reps, especially, to ask everyone to join. All right. So let's go back to the class now. We have quite, sorry for that. We have defined very well what we mean by innate immunity, adaptive immunity. So I want to believe that those two phenomena are clear. Now, we now coming into the HIV virus, okay? This thing that we fear, that we call the HIV virus, all right? Number one, we ask ourselves, how does the virus look like? And how and when does it infect an individual? And then what makes it so dangerous? You know, what makes it so dangerous that it ends up killing an individual, okay? So the first thing that we are going to appreciate, ladies and gentlemen, is the structure of the HIV structure. The HIV virus, sorry for that. And what we encounter is this, a loud-shaped thing, as you can see, a loud-shaped structure. So normally we start by drawing a loud-shaped structure, okay? This loud-shaped structure has two S on the inside. So after you draw the loud-shaped structure, you draw an S, a first S, and another S, okay? Yeah, and these two S's are simply what we call, they are nucleotides called RNA, okay? This is another strange thing that we are encountering today, RNA. But ladies and gentlemen, I want also to believe that in your schooling system, somewhere, somehow, you may have come across the word DNA, okay? DNA. And from a layman's point of view, I want to simply let you know that a DNA becomes an RNA, okay? And then becomes a protein. You know, simply let you know what happens in our bodies. A DNA gives us an RNA gives us a protein. Majority of our cells, majority of who we are proteins. You understand that? Majority of the cells that make your body are simply proteins. Okay? So all these proteins come from DNA. That's very good. We go back to the structure of the virus. So in the structure of the virus, so we have said to draw it, or rather to appreciate it, you start by first of all drawing a circle, drawing two S, and this two S simply represents the word, and remember it's in uppercase, RNA. RNA simply means ribonucleotide. Okay, DNA simply means deoxyribonucleotide. Okay, but majority of us are not biologists, so we don't go into those details. So once you draw that, then you can draw something that looks like is it a hexagon or what, what, a rectangle, and this something is the encounter is the one that enclosing the two RNAs. Okay, inside you draw this cube, this one here, this one here. I want to believe that you can see. You draw the rectangle. Okay, there we are, and you draw these two gray things here, this one and this one. So there's a gray thing here and a gray thing here. So the cube, the white cube represents something called RT. And RT, ladies and gentlemen, simply means reverse transcriptase. Okay, we are going to see it down there in the notes, reverse transcriptase. So this is an enzyme. All right, that's an enzyme. Then the rectangle, we have two rectangles in the diagrams. I hope you can see them. The rectangle simply represents another enzyme that is called integrase. And as the word simply means, integrase is to integrate into something, to join into something, integrase. And then, of course, we have the hexagons, the two hexagons. that we simply call protease. Again, these are simply protein enzymes we call protease. Okay, so we are simply doing a rudimentary structure. How do you approach in understanding the structure of the HIV virus? After you've done that, first thing, you draw the circle. Second thing, remember the RNA, 2 of them, the 3 proteins. Okay, after you've done that, now here you draw another line, and closing whatever you already have. Okay, you draw that line, and that line you call it matrix proteins. Matrix proteins. Matrix proteins as shown in the diagram. Matrix proteins. After you've done that, you can draw another line, but this one you may not draw. Whenever I was being trained, we really, because that time there was not so much, the structure was not clearly very well understood. We really drew the third one. We only drew the first one. You can draw the third one here, but of course that one I would not like you to pay so much attention to it. What I would like you now to pay attention to is these spikes, these things that look like a hammer. Green on top, that one, you can draw one there and see how they are appearing. Okay, yep, these things, they are very important. Okay, these structures. All right, and these structures at the top is called an envelope, kama bahasha, envelope protein. Okay, and this envelope protein is simply denoted with the initials GP120, and GP stands for glycoprotein, a protein that has a sugar. Okay, then the strand, the strand again is a glycoprotein GP, but in this case 41. And the protein here, the one that I told you, you draw here, this one again is called a I had already mentioned that, matrix protein. But we also have something that I left out here, a part of it, that we call core proteins, next to this structure, core proteins. You can see that clearly in the diagram, core proteins, protein 14. Okay, and that, ladies and gentlemen, simply forms the structure of the HIV virus. Most important, the outer membrane. Anyone who studies biology, they study the structure of the cell. All right, the outer membrane has what we may call, rather in matters cell, we call them cell membranes. And cell membranes consist of lipids and proteins. It's a mixture of lipids and proteins. All right, and this is what is shown here at the edge here. So I want to believe that this is clear. It's a mixture of lipids. The ones in black that looks like a hammer, that is a lipid, and the tails are proteins. They are called lipoproteins. All right, lipids and proteins. This is what makes the cell membrane of majority of our cells. So, ladies and gentlemen, in a nutshell, this is the structure of the HIV virus. Kindly pay attention. Kindly pay attention. The HIV virus consists of the RNA. All right, RNA, and I've given you the analogy. We call this the central dogma. Okay, RNA simply means ribonucleic acid. We have DNA, and we have RNA, we have protein. So in normal working of things, DNA gives us an RNA, RNA gives us a protein. Okay, but the virus is a viral RNA. It has a strand, and an RNA is a single strand. Okay, this again is biology. RNA consists of a single strand, but DNA consists of double strand. For those of us who take biology, we will introduce you into this double strand. Okay, so RNAs are usually single strands. So take note, we were doing a recap. Take note of the RNA, of the proteins, reverse transcriptase, integrase, and then protease. Take note of the core matrix, okay, core matrix protein. Take note of the core proteins, P14. Take note of the GP41, and take note of the most important, these are very important, envelope proteins, rather what we call glycoprotein 120, GP120. I want to believe that that's clear. Do we take a breather? Do we take a breather? That should be very clear now. Okay. We just have a good look at it and appreciate it so that we move on. Right. Okay. Now, and so I have explained that the viral RNA encodes structural proteins, various enzymes and proteins, etc. I don't need to repeat that. Now, this virus, we have seen that it has something called GP120 or GP120 envelope protein. And ladies and gentlemen, I want to let you know that at this juncture, that this protein is very important for the infection of this virus to the cells, to the human cells, this protein, okay? GP120. And GP120 binds to human cells. We are going to see that clearly, to human cells, that and only the cells in the human body that have CD4 receptors. Remember, I mentioned to you, I told you that we in loose language, in HIV loose language, CD4 simply denotes T helper cells. Okay, but in immunology, CD4 is a group of molecules. They are receptors. They look like that. They are a group of molecules that look like that you can find in any of the following cells: the T lymphocytes, what we call the T helper cells. You can also find them in the macrophages, in the monocytes, in the dendritic cells, and even in some nervous cells. I forgot to mention that. Even in some cells of the nervous system called microglia. I don't know how to pronounce that. Even in some nervous cells that we call microglia or microglia. Okay, you may find these cells also have these receptors. All these cells have these receptors. So what does that mean? That any of these cells, the T lymphocyte, the macrophage, the monocyte, the dendritic cell, or even this microglia is capable of binding to the HIV virus, because this protein binds to the CD4 receptor. And I've shown you the receptor looks something like an antenna on the surface of the cell. So next we are going to see how that happens. How does the virus infect the body? How does the virus infect the body? But before we do that, I want you again to remind you what we mean by CD4. Okay, CD4, what do we mean in it? I have said loosely in HIV, because I'm a biologist and I know this. In HIV, anyone who talks about CD4 or to the general public, they simply think it's just T cells. But CD4 simply do not just denote T cells. We also have cells that have the CD4 receptors, and the first category is the T lymphocytes called T helper cells, macrophages, monocytes, dendritic cells, and even the nervous system microglial cells. Okay, perfect. So we take a breather again. Do we take a breather? Go back to the public chat and see what we are 284. This is very low turnout. You don't want to be taught immunology? Jospat Congo tells us that bolding with blue ink isn't clear. Jospat Congo, you can get this. Yep, I can see from the YouTube link that's not clear. It's my error, because I should have at least used a different color. Okay, but just for Congo, these lecture materials, you are going to find them in the e-learning platform. You can download and you can follow these notes. Okay. Lewy, notes not clear. What is not clear, Lewy? I would like to know. Okay, Otieno Kennedy also tells me that the blueprint is not clear. It's because I'm using two different platforms. I'm using yep, and of course it was it was my error using the blue color. I have seen also from my end that it's not clear, but you can get the materials from the e-learning platform. Anything that's not clear, you're going to note to get it from the e-learning platform. Daniel, you would like to know whether you have tutorials after this online class. As usual, Daniel, there is tutorials after the online class. I do not know whether my colleagues can be able to cover whatever I've covered, but at least if you pop up in my class or in any other class, then I can explain this in detail, or rather you can engage me in the online platform. Okay. Okay. The colors, yes. Harriet Simiu, I have noted about the colors. But now because I've already started, I really do not wish to stop and start changing the colors. Okay. Yeah. Next time I'm going to know. Saitoti, you want to know the full name, the meaning of DNA? Deoxyribonucleic acid. Okay. I think I mentioned that. Yep. Dovino, yes, the notes are much decorated. I thought I was trying to emphasize some points, but then I have seen that some of you are not clearly getting it. But then again, I urge you, the reason why we have the online class is because I expect you to also log in into the e-learning platform, get these learning materials. We have activities in the e-learning platform, and you have to engage us in the e-learning platform. Thank you very much for those of us. Give us the code. The code for what? CCS 103? Criminally. So kindly, when you write something, just be explicit so that at least I'm able to respond to your concerns. I do not know. Cut paper haziko tayari, Thomas Mwita. Once we are ready, we will let you know, but we should be ready before maybe cut 2. Okay. That I do not know much, but yeah, I know they are not ready, but they should be ready before cut 2. I want to believe that we are going to work hard and give these cut papers back before cut 2. Okay. All right. So the slides are at most clear. So let them adjust to high video resolution. Just enjoying the class today. Nice decorated. Blah blah blah. Thank you very much. Can we be tested on the? Yes, yes. Paul Muye, you are going to be tested on the drawing. I think after every drawing, I keep on emphasizing that you have that's a reading assignment. When I mention that something is a reading assignment, it means that you have to attempt it and at least make sure that you draw. Very good. We move on, and now the next part of the lecture, we address how the virus infects the body. So the life cycle is simple. Number one, the virus infects the cells. All right. Number two, it produces DNA copy and an RNA copy. It integrates into the host genome. I'm going to explain what we mean by that. Sorry, the numbering system here is awkward. Number three, after integration. Number four, it expresses the genes. I'm also going to explain what we mean by that. And number five, it's production of the viral particles. So that's what we are I'm going to show you using something that looks complicated, but it's not complicated. Okay. And before we do this, I want to introduce you to something else. Before we do that, let me introduce you to something else. We have that. Okay. As we take a breather, let me introduce you to something else. Perfect.
Efforts to prevent, treat, and better understand. HIV/AIDS has taken the lives of over 39 million people. HIV/AIDS has taken HIV/AIDS.
So allow me to share my screen so that at least before I explain that diagram, which may look very complicated, allow me to share my screen so that at least I take you through something else. Stop sharing. Hello. So we are going to listen to a video, and this video is going to let us know, to usher us into how the infection happens. What's going on now?
HIV/AIDS has taken the lives of over 39
we
million people worldwide, despite our efforts to prevent, treat, and better understand it. But with 35 million people currently infected, what exactly is it, and are we close to a cure? To contract HIV, the virus must enter the bloodstream, and it's often transmitted from infected bodily fluids like blood, semen, vaginal fluids, or breast milk. Once inside the bloodstream, HIV targets a variety of cells, but most specifically the T helper cells, which are a type of white blood cell that play an essential role in our immune system and fighting infections. The outer envelope of HIV is covered in glycoprotein, ultimately tricking the T cell receptors to not recognize the virus. Once attached to specific proteins on the T cell, it begins to fuse the membranes together and eventually enters the cell, where it releases 2 viral RNA strands and 3 essential replication enzymes. Because HIV is a retrovirus, the RNA is transcribed into DNA, represented here by a zipper of 2 RNA strands transcribing into DNA. This DNA is then integrated into the host cell's genome. This makes the T cells treat the viral genes like their own, which causes them to make more copies of the virus. These then leave the host cell and mature, ultimately seeking more T cells. The virus is particularly difficult to treat because its mutation rate is so high. Overall, the replication process creates more than 10 billion new virions each day. During these initial stages of replication, called the latency period, a person may not show any major symptoms for up to 8 years. If not treated, the HIV eventually kills off the specific T cells it infects. When these T cells fall below 200 cells per cubic millimeter of blood, it becomes acquired immune deficiency syndrome, or AIDS. After progressing this far, the immune system becomes suppressed and is much more susceptible to cancers and opportunistic infections such as pneumonia. A person doesn't die from AIDS, they actually die from an illness that the body could not fend off. Nowadays, there is medicine that helps fight these opportunistic infections, like Daraprim, which was recently in the news when Martin Shkreli of Turing Pharmaceutical decided to raise the price from 13 drugs that slow PrEP. This works similar to antiretroviral drugs by blocking the enzyme reverse transcriptase. Thankfully, there is hope for a cure. A small population.
So, ladies and gentlemen, let's stop there now. Let's stop there. I'm going to share another video shortly after this. Let me first of all try to explain this now. All right. So, in a nutshell, or rather what that video is trying to explain in common sense, is that we have the virus here. So this is a virus, and this is what we have here, and we have a cell there. Let's put the cell in very big circle. So the virus has these things on the surface. We call them protein envelopes, GP120, GP blah blah. I hope you remember that. Okay. So, and the cell has receptors on the surface, and these receptors are what we call what? CD4. CD4. And here we have the GP120. So the first thing that happens is attachment. Okay, attachment. So this section or this structure here binds to this part, this receptor, and this receptor is what we have simply called as the CD4. So the virion, the HIV virus, which we have here, binds. The plasma membrane simply means the cell membrane, this structure of the cell here. So this is a cell membrane, or rather the plasma membrane. Okay, so you can see clearly using the diagram here that the virus is binding to the CD4 and something else called chemokine receptor. For now, simply pay attention to the CD4. Okay. After the binding, the two parts, the two membranes of the virus and of the cell fuse. Okay, they fuse. The one shown in purple here. And I hope I'm not in class alone, because right now I may be talking to myself. All right. So Salem, kindly inform me if you lose me, if I lose the connection. So after the binding, ladies and gentlemen, the fusion of the virus and the membrane. So what happens is that the virus, the two strands of the RNA you can see here, the one in yellow, are released directly into the cell. This is what we have here. The RNA virus, okay, the one that is shown with the curve there. And I remember I said it occurs as a single strand. We really do not wish to jargon, but this is what you should pay attention to. Okay, after the fusion, number one, the binding, the fusion of the membrane, and the release of the contents of the virus. These contents contained in the core matrix are released inside the cell. So here we have the RNA virus. Okay, so this virus, number one, is, so if I may go back to what I had shown you, normally, conventionally, information is packaged in DNA. A DNA makes an RNA, ladies and gentlemen, and an RNA makes a protein. So let's be guided by that. It's called the central dogma. Okay? In genetics, that is referred to as a central dogma. So this virus, number one, is transcribed back to, because we are here, the virus is here, is transcribed back to a DNA. Okay? And the molecule responsible, or rather the enzyme responsible for doing that, is the reverse transcriptase. And if I may take you back kidogo, you are going to find that we describe the reverse transcriptase in this diagram, RT, that enzyme. It's contained in the core matrix of the virus, of the HIV virus. So moving on, after making a copy of a proviral DNA, or rather what is called a viral DNA, that DNA integrates, moves to the nucleus. Now this is the nucleus that you can see here, the nucleus of the cell. That DNA integrates into the cell's DNA, okay, to integrate. And the integration, to integrate simply means that this DNA that we have formed here joins your own cell's DNA. Here we are, okay? So after joining, it now makes an RNA. So we have already made the DNA. So this is the DNA that we have made going back. This one is done by reverse transcriptase. So RT, okay? So this DNA makes an RNA, okay? It makes an RNA. So this is what we have here, the yellow one here. And this RNA, okay, makes proteins. This is what we have here. And these proteins are what constitute and are assembled to become part of a HIV structure again. Okay, so the cycle repeats. So we started with an RNA, becoming a DNA, integrating into the DNA, and the integration is done by an enzyme called integrase, which again we had already described, making another RNA, and the RNA making proteins. And these proteins are assembled together with the RNA to make a new virus. Here we are, okay? And the new virus gets out of the cell. Now, the danger is, and this is what we should pay attention, because this DNA integrates into the cell's DNA, it affects the DNA of the cell, and in so doing, it ends up interfering with the cell, and the cell dies. So the whole of this cell will die. After the virus has gotten out of it, the whole of this cell dies. I hope we are not just hearing stories or a sermon in church. Okay, we can follow that the RNA becomes the DNA. I've shown you why, because of an enzyme called liver transcriptase, which is then integrated into the host DNA. The host DNA makes an RNA. The RNA joins other proteins. RNA also makes protein, like I showed you here. These proteins are joined together to become another HIV core structure, which we started with here. This is the core structure of the HIV virus. And then the other structures are added as the virus goes out of the cell membrane. The HIV GP120 and GP41. And finally, we have the release of a new virus. So this, ladies and gentlemen, is what constitutes the life cycle of the HIV virus. And the process is devastating, because after doing this, we can see the reason why the cell has to die, because a new virus, a virus DNA, enters your own DNA and ends up interfering with your own DNA, such that if your own DNA is interfered with, that cell will ultimately die. There's no two ways about it. Okay, so the virus is released and the cycle continues. The cycle again continues. These new viruses will look for another cell because they want to multiply. Cha muhimu, and I have to talk in Swahili now. Cha muhimu about the thing about viruses is that if they are outside a cell, sometimes they live in what we may call a latent state. Okay, they are not dead, but they assume to be dead. I think in living organisms, there is something called what? In winter, some animals go to hibernate. They may assume very low metabolic states, and it's almost like they are sleeping or sleeping forever. So viruses are clever and intelligent in the sense that if they are not inside a cell, they simply assume a state of not multiplying. In fact, it's almost like they are dead. But immediately they infect a cell, they come across a cell, they are able to reselect and start making new copies of the viruses. Okay, so without going into so much jargon, if we may go back to the previous slide, this slide, we talked about the virus infecting the cells, producing a copy of the DNA copy and a viral RNA, integrating into RNA, expressing viral genes, and producing viral particles. Okay, so in a nutshell, that diagram that looks very intimidating, it's what it explains those 6 processes. Okay, those 6 processes. But then there are some little things that I would like you to pay attention to. That number 1, the reason why the virus is able to attach to the to any cell is because of these molecules, what we call the GP120, the envelopes. They bind to the molecule that we called cluster differentiation 4, CD4, number 1. Okay, then we have other cells in the cells of the immune system. Majority of them, they have other receptors. Okay, they have other receptors. And remember, I've told you, because of cells of our bodies, most of them we cannot even see them with our own eyes. We need the help of a microscope. So majority of them have so many structures and proteins on their surfaces. These are protein membranes. Okay, these protein membranes, I have used the word antennae. They are simply antennae. They are used in communication. Just like every radio has an antenna, they are used for communication. So majority of the cells that defend the body have other types of antennae. And we have 2 perfect examples of 2 antennae called CXCR4. This is an abbreviation for a protein. And this is also an abbreviation for a protein called CCR5. So these 2 molecules, again, they have very high affinity for GP120. The GP120, remember we said it simply means glycoprotein 120, this structure of the HIV virus, the one that looks like a hammer or like a sledgehammer, okay? So the reason why so many cells of the immune system are infected by the virus is because of the majority of them besides having an antennae called CD4, they also have an antennae called CXCR4, an antennae called CCR5. Okay, so these three antennae make especially the cells called T helper cells, okay, to be prone, or rather to have a very high likelihood of getting infected with the virus. And especially the T helper cells. These ones are the ones that carry the burden of the infections. And once these ones, because the virus seems to like these T helper cells because of these two molecules, especially these two molecules, and now the CD4. Okay, it bites, it's able to integrate, make a new virus, and repeat the cycle. Okay, and that the virus, now the virus is very happy because it has found a weak part that it can always capitalize on that cell and produce a new virus. Because everything in this universe, including yourself, you came to the universe because you want to survive. Part of surviving is multiplying and duplicating yourself. Duplicating yourself means at some point when you finish school, you'll need to get babies, and you'll be happy when you get your own babies and say, these are my own. So it's the same case with the virus, if I may use the analogy of living things and humans. Okay, so I want to believe that part is clear. And as we take the breather, now, because we said we are not in any hurry, we have all the time in the world for us to learn this. I would like you to appreciate another video of the infection and how it's done, just to help us. I have posted some of these videos in the. I've posted some of these videos in the e-learning platform, so you can find them there. But as we take the breather, I want you to appreciate and see a different angle of how this is explained.
So this is HIV. It's a typical retrovirus, meaning that it has an outer envelope, and in the center, it has two copies of RNA, as well as an enzyme here in blue that's reverse transcriptase, which will ultimately turn that RNA into DNA. The virus itself, with this outer envelope protein, actually directly infects T helper cells. The way that it does this is that as it comes up to the cell surface, it uses receptors that are on T helper cells and exclusive to T helper cells, which are CD4 molecule, which really defines T helper cells. It's a surface receptor that binds to the envelope protein. That causes a conformational change and allows a second receptor to grab hold of the envelope. This is the chemokine co-receptor. It's also called CCR5, and we'll talk about that more. What happens now is that the stalk of the envelope protein pierces through from the virus into the host cell and starts to draw the cell membrane and the viral membrane together. And what ultimately happens is fusion of those two membranes, and the viral genetic material is injected essentially into the cell, and the envelope protein is left at the cell surface. The virus has a matrix and a capsid protein shown here in green and red that essentially are digested when it enters into the cell. That releases the viral enzymes and the viral RNA. Here we have reverse transcriptase, which takes the viral RNA and, using host nucleotides, converts that viral RNA into a single strand of DNA. While it does that, it makes some random errors, which is characteristic of reverse transcriptase. It has very poor proofreading activity. That single-stranded DNA now is again reverse transcribed into a double-stranded DNA.
At that point, another enzyme that has come in with the virus in the beginning, called integrase, essentially grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome, and it basically, the integrase enzyme makes a nick in the host DNA and allows for HIV to insert itself into the host chromosome. And that right there is what establishes lifelong infection. Now, RNA polymerase comes along and makes messenger RNA. Those messenger RNAs encode for different viral proteins. They end up associating with ribosomes at the surface of the rough endoplasmic reticulum. And here's a piece of mRNA that's making envelope protein, which is directly produced into the endoplasmic reticulum. And it's shuttled then through the endoplasmic reticulum and taken to the cell surface, where at the cell surface, it becomes embedded in the cellular membrane. And at this point, coalescing with other envelope proteins that have been produced, you have this cluster of envelope proteins now on the surface of this infected cell. At the same time, there are other messenger RNAs that are being produced that allow for translation of other viral proteins. So here are additional viral proteins being made, which are going to be used to make up the key components that the virus ultimately is going to need. These are transported again to the cell surface, to the area where these envelope proteins are, and a strand of RNA as well as some of the enzymes are part of that complex. This then buds off at the cell surface at this point, but it's still not a mature virion because the polyprotein chain needs to still be digested into its component parts. That's done by an enzyme called protease. Protease breaks up those polyprotein chains and ultimately allows for them to coalesce and form the mature structures that make up the final virion. And now you have a mature infectious virion that can go on now to infect other cells. Once that happens now, the cell can produce tons of viruses, and this is really what then keeps the whole process going.
Thank you. Let's stop sharing now. So I want to believe that we keep on appreciating how the infection happens. We keep on appreciating how the infection happens. But then if we go back here, there are major cell types that may be infected by HIV. And I want to first of all let you know that the major cell type that is infected by HIV and that the virus the HIV virus likes to infect is what we call the T helper cell. All right, the cell that we appreciated as a T helper cell. And structurally, this is how it looks like. This is how the helper T cell looks like. The other cell that the virus likes to infect is the cell that we call a macrophage. It's a very big cell, a very humongous cell with a very big nucleus, and it usually has some granules inside it. So we normally denote it as a macrophage like that. Okay, it simply works by engulfing and eating out anything that is potentially dangerous. We call these antigens or pathogens. Okay, the other key cell that the virus infects, the other key cell of the immune system that the virus likes to infect is this powerful cell here that looks like a starfish. We call it the dendritic cell. All right, the dendritic cell. So pay attention that these three cells are the ones that majorly are infected by the virus. Okay, and remember, you may ask now, the virus is getting to me or getting to the individual through sexual transmission, through blood contact, etc. So you'll find that majority of these cells, just like we stated in the beginning, these cells are found in the blood and in majority of our fluids in the body. Okay, because they form crucial component of the first line defense. These are the major watchmen. Okay, these are the major, especially this one and this one. They are the local watchmen in every tissue in the blood. They are like the gatekeepers. They are the ones that encounter any incoming pathogens. We call them antigen presenting cells. The two cells, the macrophage and the dendritic cell. We have appreciated that the reason why the HIV virus likes the T lymphocytes is because, number one, of the molecule that we have called the CD4, the molecule that we have called CCR5 and CXCR blah, blah, blah. Okay? Very good. So we are done with the life cycle, and having appreciated the life cycle and how the virus infects the cells, then now what we need to appreciate is how then does the disease progress, the pathogenesis of AIDS. Okay, the pathogenesis of AIDS. And I want to believe that by now, majority of us, or if not all, we appreciate the difference, that there is a very big difference of what we mean by getting infected with HIV, the virus, okay, and having the disease symptoms of what we may call acquired immune deficiency syndromes. Okay, I want to believe that you may know the difference between having the virus and having AIDS. Okay, so the first thing that I would like you to pay attention is this: that over many years, once you get if I get infected today, the virus may be contained. It may be the multiplication level may be contained by the cells of the immune system. They might be able to contain it. Okay, and we remember we usually refer to this stage as latency. All right. Maybe the virus is multiplying, but at very low levels. All right. But then as the infection progresses, of course, it means that majority of the cells are dying. All right. Either whether these cells are infected or not infected, even they are non-infected. And if majority of the cells that are supposed to protect the individual dies, then it means that you are progressing to the clinical AIDS, which we are going to see later that it's accompanied by many infections. Okay, chamuhimu, and this one I would like you not to forget. Chamuhimu ikuwa that the major culprits, the major cells that get destroyed and diminished are the so-called T helper cells, or rather what we call AKA CD4 system, or the CD4 cells. And we say that these are very important cells, especially in immune responses, because like their word denotes, they are simply helps, and they simply help by communication or cell signaling. Okay, or cell signaling. Their death simply happens by a phenomenon called cytopathic effect. And cytopathic effect means if we have a cell here, and inside the cell we have a nucleus. Inside the nucleus we have chromosomes. If you interfere with chromosomes, the chromosomes contain the DNA, and the DNA is a thing that is the blueprint, rather, that controls the activities, the metabolic activities of these cells. So if you interfere with this DNA, like the HIV virus does, it means that the metabolic activities, the effects, the intracellular effects of this cell are going to will be interfered with. We call, and when they are interfered with, the cytological effects, the cytological structures, etc., they are not able to work as they are supposed to work. We call that cell having a cytopathic effect, and this cell ends up dying. Okay, ends up dying. The mechanism of dying are many. We cannot get into that at this juncture. Okay, so next, ladies and gentlemen, I would like you to pay attention to this. Primary infection. Where does the primary infection occur? In the blood, in the mucosa, in the vagina, in the penis, blood contact, in wounds, etc. Who does the virus encounter? The CD4 cell, the dendritic cell, etc. What then ensues after that is that this encounter travels to the lymph nodes. Where do we find the lymph nodes? In our armpit, in our groin, in our neck, etc. Of course, this is now in the lymph nodes. This is where now the virus keeps on multiplying. You reach a stage whereby you have viremia. the spread of the infection throughout the body. Of course, at this point or at this juncture, the body is still producing some proteins, very important proteins that we call antibodies. Okay, very important proteins called antibodies, and even very important cells called killer cells. The ones that are denoted here with CTS, we call them cytolytic T cells. They are simply killer cells that are trying to counter the virus. They are trying to come to aid of controlling this virus, but they end up being defeated because the rate of the duplication, the rate of the multiplication of the virus is too high, finally establishing what we call a chronic infection, where the virus is trapped in the chronic, in the lymphoid tissues. The lymphoid tissues, if you remember the picture, the drawing of a human being, the lymphoid tissues are distributed everywhere, and especially at geographical areas. We saw that in the armpit, there are lymphoid tissues, near the mouth, any openings, in the abdomen, in the groin, etc. In the bone marrow, we find all these have concentration of lymphoid cells, or rather white blood cells. And finally, this ends up as full-blown AIDS, AIDS, where a human being suffers from a myriad of all types of infections. We are going to later address that in the second part of the lecture called comorbidities. Comorbidities. Ladies and gentlemen, allow me to move to the next slide now. And in the next slide, I will introduce you to the viral load. And this part of this of the lecture now is a little bit quite straightforward. I'm not going to labor much on it. Viral load, what do we mean? You are infected with the virus. So if we take a milliliter of a blood, a drop of your blood, and measure the amount of viral particles in that milliliter, that is what we are referring to as the viral load. Okay, and we have parameters that have been set at more than 1,000 copies. Of course, it means that you are treading on a dangerous ground and you need to start medication. But what happens on exposure immediately after you have been exposed to the virus? Okay, number one, of course, the virus tries to multiply. It's intense multiplication between 1 and 6 months and 1 year. But of course, remember, at the same time, the body is trying to bring the virus down, okay, to a steady level, which is called a load set point, whereby at least the body is able to counter the multiplication of the virus, but it's not able to eliminate the virus. That's the danger. All right. So in advancement of the treatment of AIDS and rather the HIV, nowadays, if anyone is tested and they realize that they have the virus within themselves, then they start taking the antiretroviral drugs, and this one brings down the load of the virus. Okay, so the viral load set point, this is simply a range that is used to predict whether a person has the likelihood or the dangers or the chances of progressing to the clinical AIDS. Okay, but then if you look at the virus and the viral load and how the virus is progressing, you have to at the same time appreciate the response, because we have already seen that the virus really has an affinity for a particular type of cell that we are calling the T helper, the CD4. So this number of cells is usually monitored. Why? Because remember, we mentioned that the virus really liked this cell because of these molecules, the CD4, that molecule and that molecule. And so normally what happens, the virus destroys the circulating CD4 cells, the ones in the bone marrow, the ones in the thymus. It also destroys some cells of the immune system that have CD4. These cells are called microglia cells. Okay, and that ends up in almost finishing, or rather clearing, all the T cells, all the CD4 cells in the body, and that is a very dangerous situation. Immune depression, that is a very dangerous situation whereby these cells are almost getting diminished. Why? Because the virus is simply multiplying in them. Because if these cells diminishes or they are interfered with, they play a very important role in fight against infections. So if their numbers go to some very low levels, it means that the individual now has very high chances of contracting any type of infection. And this type of infection usually are very difficult to treat. Okay, that is the danger, and that's why the HIV virus is usually superior. It's not easy to clear it, and that's why up to now we have not had a clear-cut treatment system to rather clear the virus from the system, because again the virus is intelligent. It keeps on changing. These changes, rather in biology we call them mutations. It keeps on changing, escaping, because like I've told you, all of us in this universe are meant to survive, and all of us, we want to use resources and claim our part of the universe and multiply and make children, and then our children make children. We want to be better. It's the same case with living organisms, even viruses, the things that you can't even see with your own eyes. So normally we have a period between infection, a period that you never show in symptoms. We call that the latency stage. You rarely show any form of symptoms, and depending on individuals, again, this could last between 10, even forever. There are people who are even called long-term non-progressors, people who rarely show any symptoms, or rather who have declined in the number of CD4 cell counts. All right, people who, it's funny, and again, there are people who just get an infection today, and by December they are dead. But there are people who are rarely affected by this virus. Yes, they are infected, the virus is in them, but in the long term, they may end up getting killed by something else. They may never end up, even without taking ARVs, they may never end up having the clinical AIDS. Okay, AIDS. So during the symptomatic phase, the worst phase now, all right, this is when we have seen that this only comes about because of the depletion, or rather the deletion of the CD4 cells. And normally this is a very dangerous stage whereby an individual has so many comorbidities, so many infections, some of them are untreatable. Even cancer at this point, even depression, so many things come into being, and an individual has very high risk. If that is not managed, an individual has very high risk of progressing to death. So the first part is the acute HIV syndrome, acute, whereby patients experience, fever, chills, night sweats, etc., and this is when immediately, especially within the very first few months after you have been infected, you are most likely going to experience these symptoms. And of course, this correlates with the high number of the virus. And of course, I think I've clearly explained that in the diagram here. So if you look at this diagram, if without using a lot of jargon, if we may look at this diagram, so this is the these are the T cells in the individual on the x-axis, on the y-axis on the left. Okay, they are as high as 1,200. Okay, and on the right is the number of viral particles. They are called copies of the virus. Okay, 10 raised to power 8. You can imagine what that means. Okay, so at week zero, or at day 0, when you get infected, at this point, you can see where the cells are. All right, so as the virus keeps on multiplying, the CD4 cells are going down. Okay, and at this point, you can see this is what we are calling the acute HIV syndrome. At this point, where the virus copies in the blood is more than 1 million copies in 1 ml, in a drop of blood. I want you to imagine what that means. In a drop of blood, you have more than 1 million copies of the virus. So this is when there is widespread dissemination of the virus in all the organs, including the lymphoid organs. But then the body is also trying its way to work out and try to control. So sometimes, depending on your diet, on your exercise, on how well-being you are, or whether you are using the drugs that are supposed to manage HIV, so the cells may start rising, but then as years progress, they go down until finally the individual dies. Okay, until finally the individual dies. So this is simply how the progression goes about. Okay, high viremia, we call it high virus levels, corresponds with very low numbers of cells, of defense cells that we call CD4 T cells. Okay, so that is the main, or the rather the takeaway message from that graph, from this graph. Latency, again, remember, during latency, there may be few clinical problems. There may not be any symptoms that are affecting the individual, but then that does not mean that the virus is not in the blood. It is in the blood, and if the individual does not take care of themselves, then the virus keeps on multiplying, and this, of course, leads to the full-blown clinical AIDS. Okay, leads to the full-blown clinical AIDS, ladies and gentlemen. All right. Lastly, the last part of this lecture, part one of this lecture, is about the therapy and the vaccination strategies. So currently, as you may be aware or not aware, we don't have a treatment for HIV, the virus. All right. We have treatments for the syndromes, rather the many complications that arise out of the virus, but the virus there's no cure for the virus. And the many drugs that you hear about in the market and some of us use is called the antiretroviral therapy, ART. But these drugs simply block. Their job is they work by blocking, and they are a combination of many drugs. They work by blocking the activity of some of the some of the mechanisms of the virus life cycle, like the transcriptase, the protease, the integrase, etc., etc. So there is currently no drug to kill the virus. Okay, so we have therapies, combination of so many drugs. They are called highly active antiretroviral therapy and antiretroviral therapy. All this is supposed to try and minimize the amount of the virus, but it does not eliminate the virus completely. All right, and then the danger is the individual, like we are going to see in the second series of the lecture before we break. The danger is that the individual now develops opportunistic infections. All right, opportunistic infections. And opportunistic infections means whenever you are compromised, whenever your T cells are compromised, they cannot amply defend the body against so many, common illnesses, common cold, pneumonia, some things called skin cancer like Kaposi sarcoma, even some form of city cancers, so many complications, chest complications, TB, malaria, etc. All these things, they will find them in someone who has now progressed to the clinical AIDS. And even to some extent, even because some people, when they take this therapy, or rather the drugs, some people have side effects. So you find that some of them develop cardiovascular or rather lifestyle diseases. So ladies and gentlemen, I want us to take a breather. And as we take a breather, we do the summary of the lecture. So what have we covered today? As we take a breather, so that at least I go back to the YouTube channel. How do we access the YouTube channel? This is a funny question. I go back to the YouTube channel, and then from the YouTube channel, I want us to comment on the link on the chat. What have we learned from today's class? Number one, I want us to Kimadi, not understanding the language. Oh, yeah. I want us, first of all, to take a quick recap of what we have learned from this. We have the second part of the lecture, which will take about 10 minutes, and then we break. But of course, before we do the second part of the lecture, I want us to, first of all, recap. What have we learned? I know the last part I've tried to be a little bit faster, Otieno Kennedy, but then what have we learned? Number one, have we appreciated the structure of the HIV virus? Have we appreciated the structure of the HIV virus? That's the first thing that we need to ask ourselves. Have we appreciated the structure of the HIV virus? Okay. Do we know the cells of the tissues, and the molecules of the immune system? So that's the second thing that we need to do a checklist. We do a checklist of what we have covered in this part of the lecture. All right. Do we appreciate, I hope you are writing or noting down, do we appreciate the distribution of tissues of the immune system? The distribution of tissues of the immune system. Kindly type in the public chat so that at least I know whether we are together. Do we appreciate the second thing that we have learned today? Can we appreciate the cells of the immune system? The ones that we find in the blood, the one that we find in the tissues. Do we appreciate them? And this forms part of the reading assignment. From this lecture, can we be able to explain to someone or to our brothers or to our parents what we mean by the CD4 cells? Rather, not just by what we mean by the CD system, but rather what we mean by the CD4 cells. Okay. Can we name the two arms of the immune system? Can we name the two arms of the immune system and what they do and how they protect us? Right? Can we explain to someone how the virus replicates? How the life cycle of the virus? How does the virus infect the body? Can you explain to someone how the virus infect the body? Can you explain why the virus likes the CD4 cells, rather what we call the T cells, the T helper cells? Can we appreciate, or rather, can you explain why the virus likes that cell? Are we aware of the major cell types that may be infected by HIV? Are we aware of the major cell types? We have mentioned them. The major cell types that are infected by HIV: the T lymphocytes, the macrophages, the dendritic cells. Okay. Are we aware of that? Right? Can we explain how the disease progresses from the initial stage of infection to the progression of the disease? What we have just explained, the pathogenesis of the disease caused by the HIV virus. Can we explain that? Can we explain to someone what we mean by a viral load? Okay, what do you mean by a viral load? What happens on exposure to HIV infection? If I get exposed to the HIV virus right now, what happens next? Can you explain to your mom or to your dad what we mean by CD4 cell count? What do we mean by CD4 cell count? Are we aware that when some people get infected with the virus, they rarely show any symptoms and they can survive for the rest of their lives without ever showing any symptoms of AIDS? The so-called long-term non-progressors. Are we aware about them? What happens? The next thing is what happens during the symptomatic disease and AIDS, what happens? What is a person likely to go through? Which infections, which symptoms are they likely to go through if they have symptomatic disease and they show they progress to the clinical disease? Can you be able to define what we mean by latency? Latency. Can you use a diagram and explain or rather demonstrate the clinical course of HIV disease? Can you be able to use a diagram or even a graph to explain to someone using very simple terms that the clinical course of HIV disease is monitored using two parameters, the viral copies and the CD4 T cell count? Can you use a diagram and at least show someone at this point of infection, an individual usually have around 1,000 CD4 cells. By the time they die, their CD4 cells are less than 200. Okay, so ladies and gentlemen, I want to believe that that part of the lecture is okay. If remember, there are things that because we are doing an online class, not a physical class, there are things that you have to refer to the diagram, to refer to the notes and read. Again, in the interest of time, we cannot have, I cannot be able to attend to all the things that I would wish to cover in the online class. But then allow me to take 10 minutes to explain something. And then, of course, I will ask you to do an assignment. Okay? Just 10 minutes to explain something. And then, of course, allow you to do an assignment. So, ladies and gentlemen, now that we have seen, the virus, how does it infect the body? How does the disease progress? All that. We have explained that. Okay? Now, we need to understand that when we talk about pathogenesis, okay, then we have another term we want to bring into called comorbidities. Sometimes I have a problem pronouncing that, comorbidity. Okay, and so what do we mean by comorbidity? This one is going to take less than 10 minutes. What do we mean by comorbidity? Okay, now I have already mentioned that, and it's not good to say I have already mentioned. I want to bring to your attention that anyone who has progressed to clinical HIV/AIDS, they suffer from a myriad of infections. We have seen pneumonia, tuberculosis, diarrhea, fevers, chills, malaise, depression, etc. Name them. So comorbidity simply refers to when an individual is suffering from one or more illnesses at the same time, either simultaneously or sequentially, one after the other. And sometimes you are not able to explain why. I don't know whether you understand. Because if today I'm suffering from typhoid, tomorrow I'm suffering from malaria, and the next and the following week I'm getting treated for pneumonia, then it shows that there is something wrong in my defense mechanism, because normally that is not how we are created, and that's not how we are structured and wired. Okay, so the term comorbidity simply implies the interactions between the illnesses that affect the cause and even the diagnosis of all these diseases. And the reason why we have to emphasize on this is because in situations whereby an individual has now progressed to the full-blown AIDS, they suffer from all these illnesses. And sometimes these illnesses generate other illnesses. And this leads to mortality and sometimes to, quick death. Let me just mention quick death, not just mention the other jargons that I wanted to mention. But what we should appreciate is that the commonest things that take people to grave those who are suffering from HIV, not suffering rather, because some of us are born with HIV and it's not our mistake. All right. Remember, we say this in class many times. If you are born with it, then it's none of your business. You continue taking the drugs like nothing is happening. Okay, and you live your full life, and you should not even be worried about what other people are talking about. You take the drugs and you are assured of not suffering. You just die at your old age. So, but the commonest things that take people to grave because of HIV are listed from number A to H, from diabetes, hepatitis, kidney disease, depression, even drug use. Some people fall into depression. You may ask, mental illness. Why? Because they are not able to accept that, number one, they are infected with this virus. All right. And I've explained of situations whereby someone is born. I mean, you inherited the virus from your parents. There's nothing you can do about it. And so if you reach a point whereby you keep on denying that, oh, I cannot take the drugs because blah, 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 I mean, you simply sending yourself to an early grave. Drug use, people use drugs when they are trying to run away from problems. And drug use comes with the danger of addiction. Addictions has its own problems. All right. So majority of, majorly what I've listed here is the majority of what has been documented as the major comorbidities. But then anyone else suffering who has not even progressed to the full-blown HIV disease, but they are HIV positive, they have higher chances of contracting rather liver diseases and kidney diseases. Their kidneys most likely are going to fail. Most likely they might suffer from syphilis, gonorrhea, etc., etc. Most likely, again, they might tend to use to have psychiatric disorders, or even to abuse some drugs. So this is documented because in medical terms, this is things that the medics advise, that the people who are diagnosed, or rather found, or rather have been tested to have HIV positive, they tend to run away from the existing situations, and sometimes try to use maybe drugs or other things. Some people even go to an extent of revenging. You have heard in the news some people bragging, I have infected 101 people because I mean someone infected me knowingly, so I went ahead and revenged. And I want to believe that anyone in my class will never go to an extent. I also want to believe that if you are in my class, you can never go to an extent of refusing to take drugs. If you refuse to take ARVs, then you are simply trying to tell us that you want us to bury you next year. And we emphasize that you are in our classes because we want to mold you, and you sit wherever we are seated when we are old, or when we leave this world. So then the most important thing to understand is that we simply categorize, and I really do not like this, we simply categorize anyone living with HIV as a person living with HIV, PLHA, all right? And the people who undertake measures and go to an extent of even taking drugs, living a good lifestyle, exercising, etc., taking care of their mental health, they die from age-related chronic illnesses. They never die because of AIDS. They never die because of AIDS. And so these comorbidities, they have their own consequences, like diabetes. Of course, diabetes, we have the two types. We are not going to go into the details, but the commonest one is the one that deals with the blood sugar, and it has very serious consequences, especially when left untreated. And generally, anyone with HIV have higher chances of becoming diabetic, all right, or rather being unable to control their blood sugar. Same case with kidney disease, but kidney disease comes in twofold. You could be taking drugs, and the drugs are taking a toll on your kidney, because your kidney is the filter of your blood. The kidneys, anything that you consume with your mouth, it is absorbed in your blood, and then from your blood, it leaves your body through the kidney. So the main filter, the major filter in your body is through your kidney. And much of the time is that sometimes the virus affects the kidney cells, but majority of the times is when the virus, the drugs that are used to manage HIV interferes with the filtering system of the kidney. High blood pressure. For the high blood pressure and cardiovascular diseases, mostly these are associated with lifestyle, like smoking, lack of exercise, and of course, the ARTs, the ARV drugs, okay? The ARV drugs that majority of the patients are taking. Hepatitis C and hepatitis B, all right? Hepatitis B and hepatitis B, but these ones mostly arises because of the low immunity. The majority of these people contract these two hepatitis because of the low immunity. But the danger is these viruses sometimes are associated with liver cancer. So again, it's important that you take care, you eat well, exercise, take vegetables, blah, 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 blah. Depression, drug use comes because of, not accepting the situations that you are in, all right? If you deny that I cannot be infected, and I've even mentioned that some people deny to an extent of anger results to revenge, you go infecting people. Remember, surely you should even be jailed if you infect someone knowingly, okay? Yeah, you should even be jailed. So again, these things, anything to do with drug use and depression, it has to do with your mental well-being. So at any one point, even those of us in campus, if you are living with HIV, if you are taking the drugs, always do not go to the point of revenge. Even if the virus was transmitted to you by someone who knew whatever they were doing, or maybe by accident, it's not your, do not spread it. Do not, because some people will say, why should I die alone? All right. Remember, there is no joy in seeing someone else suffer. And remember again, just be a responsible, do unto others as you would wish to be done unto you. But again, remember, all sex has to be protected. Practice protected sex. That's the message that we emphasize in my class, that all sex in campus or in your hostels has to be protected sex. We have so many condoms, dispensers in our washrooms. So make use of that, all right? As I end the lecture, ladies and gentlemen, I want to emphasize that, and this is the take-home message from the much jargon that we have learned today, that people don't die from the virus. People don't die from the virus. You may know so many people who are infected of the virus. I personally know many of them. Some of them are my students, colleagues, family, friends, etc. But people don't die from this virus. People die because of other complications that arise from the disease. And the people who live long, the people who don't die, who die at their old age, they take care of themselves, number one, by eating well, dieting, and exercising, etc., etc. And number two, taking care of their loved ones. And majority of them only die because of other complications and not because of the virus. So in as much as we are preaching the gospel of protecting yourself, blah, 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 how about the person who is already infected? Remember, this is not a death sentence. You can't die from the virus. You can defeat the virus, and the way to defeat the virus is simply taking charge of your life, from what you eat, how you think about yourself, and how you lead your life. Ladies and gentlemen, I want to end the lecture right now. I see it's 8:54. We have been here for 1 hour, 54 minutes. And now I'll go to the public chat. And as I do that, I will take a breather. I'll try to respond to some concerns in the public chat. And as I do that, again, you can be on your way to the class, because your respective lecturers expect to meet you there. Thank you very much for joining my class. It's been my pleasure. And kindly subscribe to my channel so that we learn more immunology and we enjoy enlightening one another. So I'm back to the public chat. I will be I'll be responding to some few concerns, the ones that I can be able to handle, and then I wish you a very good day. Remember, revise. There are so many things, there are so many activities in the e-learning platform. You have to attempt them. There is no shortcut in passing. And remember, like most of you are telling me, why are we taking this course? This course, what is the importance of this course? Remember, well, it may not be very important for you, but at least you need to be informed of what is involved and how the infection happens. So we don't wish to make anyone's life difficult. Simply follow our instructions, pass the course, and move on with the rest of what you are supposed to cover in your different specialty. I wish you well. Have a good day. Any question, I'm going to try to answer it now in the public chat. You can now start dispatching to your respective lecture rooms and meet your lecturers. Thank you very much, and have a very good day. Joshua Olengo, I repeat everything, please. Joshua, we don't have time for repeating everything. We can only meet in a class, and I'm able to repeat. Okay, thank you, Bilton. Thank you, James. Kailemia, thank you. Have a good day too. You can download all these lecture materials from the e-learning platform. Kindly visit the e-learning platform, attempt the activities, and do whatever is required. All right, thank you, Bridget, Brighton, Wandela. Thank you. You can now dispatch. Please be dispatching. It's 8:56, and I expect that your lecturers are waiting for you at 9, and so that at least you meet them and they can make some. Is it true that with time ARVs adapt to the immune system? ARVs do not adapt to the immune system. It's the virus that becomes resistant. Paul, that's a very interesting question, but the ARVs do not adapt. The ARVs are not designed for the immune system. They are designed to counter the replication of the virus, okay? And so they don't add, Hussein Amuka, Amuka should be my classmate. Amuka utosheke na CXCR5 and CCR4. Your station, that one is for biologists, okay? These are simply protein names. Let's not go into the naming system. The initials are enough for that. I have tried to filter that so that at least we are only now that you are using your own where you teach immunology. Tabitha Kimodo, I do not understand that question. You may come to the Department of Biological Sciences. I can teach you some more interesting things. Okay, have a good day, William. Good day to all of you. I see 120 of you are still here. If you don't have any question, then you can dispatch to your respective classes. Yes, there is some assignment. There are some activities at the end of the lecture. If you refer to the e-learning platform, you're going to find some e-learning activities rather, and you should attempt them. Okay. Thank you all. Thank you. Dispatch for your respective lectures and meet your lecturers. Yep, Patrick, Hilary are finished. Thank you, Dennis. Thank you. The most important thing is these things are not difficult. If you revise, you're gonna pass. Okay, how does the different classes of ARVs act to slow down the procession of the virus at different stages? That is fission, reverse transcription, and integration. Phineas Kaberia, we discuss that later. The different classes of ARV and how they work. We may not go into those details for now, but they simply inhibit. The molecular mechanisms, Phineas Kaberia, that is not for this class. The molecular mechanism of how a particular ARV blocks the activity of an enzyme, we cannot go into the details in this class, okay? in as much as we would like to. They are interesting things, and some of you who may wish to proceed with this, we can always have a chat. Okay, they are very interesting things for us to learn, but of course not at this juncture and not for the scope of this lecture. Daniel Mwanza, I wish you teach us all other online lectures, sir. You are perfect. Thank you. Thank you, Daniel. Okay, Sam says Group D, they are not meeting. Thank you. So Group D, you are not meeting. Idris Bolu, thank you. The rest, kindly dispatch if you do not need to be in class right now. I want to end the lecture now. I want to end the meeting with your permission. Dr. Nyabuga, Dr. Gidaiga Selem, I want to end the meeting. I want to believe that it's not premature if I edit at this juncture. Dr. Nyabuga, Dr. Gidaiga, and Selem. Kwani niko peke yangu? Dr. Gidaiga